third AHA Allen Initiative Team Lead, Mukesh Jain from University Hospitals Cleveland Medical Center, will be our next speaker, optimizing the function of a redefined neurovascular unit to prevent age-related dementia. Mukesh.
All right, good afternoon, everyone. Thank you very much, Kathy, and to the Allen Frontiers Group, and I also want to thank the American Heart, and I think Dr. Rosemarie Robertson somewhere in the back. There she is. Thank you, Rose, for your support. So I'm going to be talking about restoring the neurovascular unit with a particular focus on the vasculature, and so it'll sort of dovetail well with Tony's talk as well. I'll begin with just an overview of things that I think certainly an audience of this capability understands well, but just to underscore the importance of age-related dementia. It is an enormous source of morbidity, mortality, and healthcare expenditure within North America and worldwide. The numbers, as you can see here, the lower number is 2015, and by 2050, the numbers across the globe, as well as in our nation, will triple. Dementia is a broad term with diverse causes. Alzheimer's, of course, is the one that is most commonly discussed. But as you heard from Dr. Weiss-Corey, the vasculature is also a major cause, and it can coexist with other forms of dementia and exacerbate disease. So the AHA Allen Initiative was really, I think, just a brilliant effort to try to get individuals that are interested in the blood vascular system and the brain and get them together to try to look at new ideas, identify new mechanistic insights, actionable insights for therapeutic gain. I should have apologized for the group. I'm a little under the weather, so you're going to hear me be hoarse, and I apologize if I sniffle here and there. So we put a team together of experts from 3 institutions in Cleveland. At Hopkins, Dr. Saul Snyder, shown for you there. In Cleveland, yours truly, Dr. Andrew Pieper, who's co-director, Jonathan Stamler, James Reynolds, and then from Penn, Zoltarani, Rod Jain, Mark Kahn, Dr. Trojanowski, and Alice Chen-Plotkin are translational scientists who have enormous access to patient-based samples, blood, CSF, tissue, and closer to our home in Cleveland, Alan Lerner, also a leader in Alzheimer's disease nationally. And we put together an advisory board of cardiovascular, blood, and neuro leaders and aging experts from across the country to advise us. So I'm going to set up the comments that'll follow over the next 2 slides. And this is a very important point and remarkably not necessarily well appreciated even amongst neurobiologists, which is that the brain has a voracious appetite for energy. It constitutes only 2% of total body mass, but consumes 20% of energy. Yet, curiously, the brain lacks its own reserves. So that continuous demand for energy is supplied by the vasculature, and in particular, small blood vessels. I'll refer to them as small blood vessels or capillaries interchangeably. These small blood vessels are about 75% of the surface area of the entire vascular tree, and they are, if you will, the business end of the vasculature, for it is here that provision of nutrients and oxygen is provided to cells of the brain. So nature was very smart to anatomically and functionally couple small blood vessels and brain cells into a unit that you're all familiar with, the neurovascular unit. And the cells proper for the vasculature are the endothelium and the pericyte that provide support and many other important functions. And then, of course, you have the various cells of the nervous system, neurons, astrocytes, microglia, etc. However, and this is an important part of our proposal, we believe that the red blood cell is an important and missing element in prior studies that have looked at the neurovascular unit, and I'll try to make the case to you that the red blood cell endothelium, that unit, what we refer to within our group as the core vascular unit, governs blood flow and consequently the provision of oxygen and nutrients to cells. The importance of small blood vessels is shown for you here in this rendition of the capillary network. You see how heavily invested the head region is. Red blood cells, I remind you from basic medical knowledge, that red blood cells in capillaries line up single file like soldiers, and they interact with endothelial cells, and there's enormous crosstalk that occurs over thousands of miles of vascular highway every second of life. And so the central hypothesis for our work is that the most abundant cell in the body, the red blood cell, and the largest organ, the endothelium, together constitute the key functional unit or the core vascular unit that governs blood flow, oxygen nutrient delivery, and paracrine signaling to maintain health of tissues. And if you're focused on the brain, obviously a readout would be cognitive function. And it follows that loss and or dysfunction of red blood cells, endothelium, or both, would impair blood flow, the provision of energy, and potentially exacerbate age-related dementia. And I'll show you some preliminary data in that regard. In addition to the focus on those cell types, the red blood cell and the endothelium, there are molecular pathways that constitute discoveries from team members within our group. And I'll focus, as I walk you through the data, on the red blood cell and its release of gases like nitric oxide in a form called snow or hydrogen sulfide. These gases transmit signals, hence they are called gasotransmitters. They impact certain molecular pathways within endothelial cells, and our focus is on two, KLFs and the NAD pathway, and specifically sirtuin proteins, and that there is a mutually agonistic relationship between these pathways that leads to a self-reinforcing molecular loop that maintains health and health of the vasculature and allows for the supply of blood flow and energy to maintain tissue health. We have three objectives. Objective one, and I'll go into more detail, so I'm going to go real quickly through this slide. Objective one will focus on the red blood cell. Objective two on the endothelium. And objective three leverages these insights for therapeutic gain. So, objective one focused on red blood cell and those gases. Some of the critical questions that we're looking at is, does the ability of red blood cells to carry these gases change with time, be it in animal models or in human subjects? And does manipulation of the ability of red blood cells to carry these gases impact age-related dementia? Does it impact the function, the RNA transcriptome, the proteome, and other aspects of other cells within the neurovascular unit? So just changing the delivery of the gas in red blood cells, does it have both biochemical and functional consequences on cells of the neurovascular unit? And so the background for this actually is very important to understand, because this is fundamental physiology that arguably, if you accept that blood flow is about as important a physiologic process, this I think is important to understand. So studies dating back to the 1800s, late 1880s, suggested that blood flow is very tightly coupled to metabolic demand. The better part of a half century later, Arthur Guyton and John Ross would show that low oxygen regulates blood flow. And they would go on to show that the most abundant protein in red blood cells, hemoglobin, its ability to hold oxygen, or its saturation with oxygen, not only regulated oxygen delivery, but regulated blood flow. why would hemoglobin, a protein carrying oxygen, regulate blood flow? That question would be answered a few years later by team member Jonathan Stamler, who would identify hemoglobin to carry a third gas, nitric oxide, which by this time had been appreciated as an agent that could dilate blood vessels. This nitric oxide is bound to a cysteine residue on the side group, the thiol side group, and it carries NO in a form called SNO, or S-nitrosylation, or SNO for short. Hemoglobin donates SNO to dilate the blood vessel and optimize both nutrient and oxygen delivery. And these efforts would really change our perspective of the respiratory cycle. The importance of this can be seen in now unpublished studies where you mutate that critical cysteine residue. So residue 93 is changed from cysteine to alanine. It is no longer able to carry nitric oxide in the form SNO. And these animals now in muscle tissue have reduced ability to enhance blood flow or maintain blood flow in the setting of a hypoxia. This is the mutant in green, cysteine to alanine, and this is the wild type. And this is quantitation for you. In very recent data in collaboration with Andrew Pieper's laboratory, Stamler and Pieper show that these animals at a relatively young age start to develop evidence of neurodegeneration and blood-brain barrier permeability. And so cognitive studies and blood flow studies in the brain are the next steps, but these are pretty good data and suggestive of the importance of the ability of hemoglobin in the red blood cell to deliver NO as being critical in maintaining a healthy brain. In addition, some years ago, Saul Snyder's laboratory identified another gas, hydrogen sulfide. It too is carried on red blood cells. It's also produced within many cells, and there's an entire enzymatic machinery that controls the production of hydrogen sulfide within cells. It is also important as an oxygen sensor and vascular relaxation and control of blood flow. Hydrogen sulfide also confers neuroprotection by virtue of a whole diverse set of effects that are tissue protective. Its levels are reduced in a broad spectrum of neurologic disorders. And in work that was just published actually this week, or accepted for publication in Cell Metabolism, they've shown that with age, the enzymatic machinery that regulates hydrogen sulfide is reduced with age from 1 month to 24 months. The amount of S proteins that are S-sulfhydrated is reduced, and these animals demonstrate cognitive impairment in the kind of testing that Dr. Weiss-Corres shared with you moments ago. So that is the red blood cell gasotransmitter component. In AIM-2, you'll recall I said we're going to focus on the endothelium and two specific pathways, the KLFs and NAD. The levels of these are reduced with age within vascular cells and within endothelium in particular. I noted this for you, but I remind you that these gases can actually augment the levels of KLF and NAD, and I'll share with you evidence for that in a moment. Some of our sub-aims here are these pathways altered, and if they are, do they impact ARD? What is the nature of this crosstalk between these molecular pathways and the gasotransmitters? How does this occur? And then, of course, are these pathways also altered in human disease? So before I share with you some of the both published and unpublished data, let me just again give you a little bit of background and a historical perspective. So the endothelium, the inner lining of blood vessels, has been the source of great interest to cardiovascular biologists for, frankly, centuries. Virchow in the mid-1800s noted that irritation of what he called the arterial endothelium, what we today call our arterial intima, which we refer to now as the endothelium, that irritation of this might be important in human vascular disease, and he based this on human pathologic samples of atherosclerosis. A century later, not too far from here, Gimbroni and Folkman, as well as in New York, Jaffe and Nachman, would forever change vascular biology and usher in the modern era by virtue of their ability to advance methodologies to culture endothelial cells. This was very important because even back to Virchow and over the century that followed his discoveries to that of Jaffe and Gimbroni, the endothelium was thought to be an inert cellophane lining, which if injured would lead to processes that would impact the blood vessel. But these efforts showed that endothelial cells were viable. They could proliferate, they could migrate, and carry out metabolic functions. In the 1980s, the importance of the endothelium took an enormous leap forward with the work of Furchtgott, showing that if you denude the endothelium, you markedly abrogate the ability of blood vessels to dilate. And colleagues Ignaro, Moncada, and Farid-Murad would identify this endothelium-derived relaxing factor as nitric oxide. And then a stone's throw away from where we're all sitting, Peter Davies and Mike Gimbroni would show that the endothelium was responsive to blood flow. The endothelium was required for optimal blood flow, that was well known, but it was also responsive to blood flow. And, let's see, I think we just froze. Let's see if that works. Oh, good. And then some years later, in the early part of the last decade, my laboratory would identify a family of factors that were flow-inducible, that would regulate nitric oxide production in endothelial cells, and we would expand that over the course of the last 10 years or so to show that KLFs, particularly KLF 2 and 4, the numbers are not so important, but they're enriched in endothelium, regulate a broad spectrum of cardinal endothelial functions. Recently, we published evidence really proving this in an adult animal. And what we showed is that inducible postnatal deletion in an endothelial-specific fashion of KLF2 and 4 results in animals succumbing within days of the levels being reduced. And what did they die from in these double knockouts? They died from small vessel hemorrhages, be it the brain, be it lung, frankly, all tissues. These data really demonstrated that there is an absolute requirement for these two factors to maintain an intact vasculature, oxygen nutrient delivery, and survival. If you lose one of the two factors, the animal actually survives, but it is not healthy. And a single knockout in both published and unpublished data, we have good evidence now that single knockout of these animals as they age, they begin to develop atherosclerosis, either to a provocative stimulus or even a low level just with age, heart failure, aneurysms, pulmonary hypertension, classic diseases that increase as we age. With respect to the brain, we've shown that endothelial loss of KLF4, the same can be said with single loss of KLF2, that these animals demonstrate impaired cognitive function. There's two tests shown for you here, the Maze test that tests learning and memory, and then motor coordination, both reduced in red, the knockouts. The brains demonstrate evidence for endothelial degeneration and leak. You see red blood cells outside of the capillary proper. And conversely, if you have transgenic animals and age them, these are animals that are aged to 2.5 years of age, they demonstrate improved cognitive function as well as motor coordination. The second pathway that we're focused on really is an outgrowth of work from one of our team members, Zolta Rani, who with colleagues demonstrated that the NAD pathway, well known to be reduced with aging, it's also reduced in the endothelium, that reductions in NAD and consequently sirtuin function are very important in reducing vascular function and consequently exercise capacity in animals. Critically, they would also demonstrate that one of the gases, hydrogen sulfide, augments NAD to control endurance in animals, and that NAD mimetics could augment exercise, a classic phenotype that occurs with aging. And so Dr. Irani and colleagues will now be exploiting these observations and studying them in the context of brain vasculature. And then finally, bringing it all together, last couple of slides on translation really stem from the work of Andrew Pieper, who's in the audience, and James Reynolds, who is not. But both of them are very interested in translating these discoveries for therapeutic gain, and I'll go through some of their observations for you now. So Dr. Pieper's laboratory a few years back, in what I would describe as a very courageous screen, identified or studied thousands of compounds in vivo looking for neurogenic and neuroprotective activity, and would identify a factor that they called P7C3 that has incredibly protective effects across a broad spectrum of neural diseases. And they linked this to the ability of P7C3 to augment NAD levels. More recently, in unpublished data, his laboratory has shown that P7C3 augments endothelial expression of KLF2, KLF4, and a classic endothelial nitric oxide producing factor, ENOS. And so the importance of this compound are now being tested in models of age-associated dementia. And then the final is RBC enhancing therapies. I spent a lot of time talking to you about nitric oxide. So Stamler and Dr. Reynolds, shown for you here, have also shown in a whole variety of disease models, this is peripheral artery disease, that the levels of red blood cell SNO carrying capacity are reduced. They developed a stable form called ENO that can be inhaled and is very reactive and can add SNO onto hemoglobin. So excited are they about their work that Dr. Reynolds, shown for you here, team member, is actually the first patient to receive ENO. It's very Barry Marshall-esque of him. And this is his oxygen saturation. As he received higher doses of ENO, his oxygen saturation improved to nearly normal levels. So this is now entering phase 1 clinical trial, and I was remiss to note that P7C3 that Dr. Pieper identified, we hope will enter human testing under the auspices of this award. And so with that, I'll summarize and leave you with a few final thoughts. Small blood vessels are requisite to meet the brain's enormous metabolic demand. The importance of endothelial and red blood cells, a core unit that governs blood flow and energy delivery. Key pathways that I've emphasized for you have been identified, and they are being studied both mechanistically and being targeted therapeutically. And with that, I'll stop and again thank the Allen Frontiers Group, the American Heart Association, and all of you for your attention.